this slide is about enrichment media. So enrichment media are used for fastidious organisms. So these kinds of organisms are, we call them picky eaters. So it's things that need, it's bacteria or other organisms that need something extra in the media in order for them to grow. So just like a normal tryptic soy agar doesn't work. So they have special nutrients in them. So it doesn't suppress anything. It's just, not everything will grow on it. So everything grows like crazy on it. So let me try to find the pointer here. These are blood agar plates. So they're sheep's blood agar. They have 5% sheep's blood in them. On here you can see sometimes the blood is actually totally degraded by the organisms, so it looks clear around them because they totally break down the hemoglobin. Now this media, wait a minute, sorry. The media that's over here is, well, shoot, I can't find the pointer. The media that's over here, this is actually chocolate agar, and it's not really chocolate, it's actually just blood that's cooked. So the blood is autoclaved, so it looks, they think it looks like milk chocolate, but everything will grow on it like crazy. So no big deal, but these are enrichment media. We'll use these at the end of the semester to grow strep. Okay, now methods for cultivating organisms. So when we're growing organisms, most of the time our goal is just to get one colony of an organism. So we want single colonies. And the reason we want these is because one colony comes from one cell. So a colony, so if I draw a colony should be, if that's my lovely colony, a colony should be copies of the cells, so it should be clones of them. They should all be exactly the same. So now when we count colonies, the units we use to describe them are CFUs, and CFU stands for colony forming unit. So CFU. Colony forming units, well, I'll show you when we go to count them, but colony forming units, that's just how we measure them. Instead of using grams or milliliters or something like that, they use a colony forming unit. So I have three methods right here. So pour plate method, spread plate method, and streak plate method. We'll do the bottom two as the semester goes on. Okay, so I have a couple things on this slide. So this slide is to explain how we do serial dilutions. So here's serial dilutions. So serial dilutions, let's see. I'm trying to find a color. No. Okay, and I'm going to get a pointer. So serial dilutions, we'll use the lightsaber because it's just so silly. Serial dilutions are how we measure concentrations of bacteria because you can't really count them. Okay, so if you looked at something that had 50,000 bacteria per milliliter in there, that's a lot of bacteria, so it'd be really, really hard to count that many. So what we do is we dilute them out. So we take test tubes with 9 milliliters of water in each of them, so 9 milliliters plain. And what we'll do is we'll take 1 milliliter of the original culture, and then we'll, the Star Wars thing's starting to bug me. Okay, 9 milliliters of the original culture, and then we'll put it into the next one. Oh wait, so we have 9 milliliters of water. Okay, and we're going to take 1 milliliter of the original out and put it into the next one. So that gives us a 1 to 10 dilution. Then we'll take 1 milliliter out, put it in the next one. That gives us 1 to 100. We'll take a milliliter out, put it in the 9 mils of water. That gives you 1 to 1,000. and then you take a milliliter out, put it in the last one, and then you get a 1 to 10,000. So from this, it makes it way easier to count the number of bacteria in there rather than the 50,000. Now, in order to count it, what we're going to do is we're either going to do a pour plate method or a spread plate method. And we'll do a spread plate in lab this term. So I'll go over this in class so you make sure you understand it. Okay, now what you do for this, or we can use that. Okay, for the spread plate method, it's the easiest. So all you do is you're going to take a sample out of those dilutions and you spread it, you pipette it on top of the agar, and then you use a little glass, a sterile spreader, and you spread it out over top of it, and then you turn it upside down and you incubate it, and the bacteria will grow on the top, and then you can count the number of colony forming units that you have. Now, pour plate method, I think it's tricky to do because the idea is you put the sample from the dilution in the plate and you pour the media on top of it. And the media has got to be like exactly the perfect temperature for this to work. If it's too hot, it'll burn, kill the cells, and if it's too cool, it'll get solid too fast. So then what you do is you turn it upside down, incubate it, and then you can count the colonies. This is tricky. I think this one's hard to do. Okay, so here's a pour plate. So what you can hopefully kind of see is some of the colonies are on top of the agar, some of them are down beneath, some of them are inside. So it's not, everything's not growing on the surface. The disadvantages, it's hard, that's not what I want. The temperature, it's hard to get the temperature right of the agar. Not everything will grow deep in the plate. So it's just really not, it's not easy to do, and it's not, I don't know, it's not very fun. Okay, not that everything has to be fun, but it's hard to do. Okay, now one method that we will use, like after this week, we'll use it almost every day in lab, is called streak plate. So streak plate is the idea that you take your loop, okay, so you take your sterile loop, you flame it or incinerate it, and then you go over and you make like an S or so on the plate. And then you flame your loop again, and you make another S, and they have to be touching. Okay, then you flame it again, and you make the third and final, like S or Z or whatever you want to call it. So the idea is you are diluting these out without having to use water. So when you touch it, and you flame it, and you touch it again, you're diluting the bacteria out every time you streak it. Or, yeah, every time you streak it. So if you look at this plate, okay, so this was probably the first one they did. That was 1. Then they flamed it, and then they did number 2. And then they flamed it, and they did number 3. And this one, they have 4. So they flamed the loop again, and they did a 4th streak. And the goal of all this is to get single colonies. So here you can see they have individual colonies. So they've isolated the organism, which is the big goal. Okay, now here's one that has, so this is the plate, and then this picture is magnified. So this plate has got 2 different organisms on it. One has got the red one, which we looked at, and the little yellow one we looked at when we were gram staining. So their goal is to separate them out. So you could pick a yellow colony, and you could probably pick a red colony and get 2 plates. You could isolate from here and get 2 plates that with pure bacteria. And we're going to do it a whole bunch of times. So if it doesn't make sense like this, it should make sense when you go to do it. But that's a streak plate. Advantage is it's just really easy. It's not hard. It only takes one plate, and you just have to flame your loop. Okay, now, the growth curve. So whenever we grow bacteria, everything has this is the typical growth curve. This is what it's going to look like. So on the x-axis, whoops, sorry. On the x-axis, we have time. Okay, and that could be hours, it could be minutes, it could be days, depending on the organism. And then on the y-axis, we have the number of cells. Okay, so here's the curve. So if we start at A, the first thing that we have is actually, lag phase. See, I don't know if this will work. So this is lag phase. So lag phase is where the bacteria are just put into the media or put onto the plate, and they're getting used to it. So they're really getting accustomed to their environment. Okay, now the next phase that we get is log phase. And in log phase, what happens is the bacteria are growing and dividing, and they call it exponentially. So that means everything is doubling really fast. So it means everything is good. They have plenty of food, plenty of, that's supposed to be dividing, plenty of space, plenty of oxygen. Everything is going really good. So they're just growing and dividing like crazy. Okay, so this would be young and growing. This might be like adolescence. Okay, now at the top here, okay, this is called stationary. I always think of it as a plateau. So basically what's happening in this phase is death of cells is equal to the growth of cells. So death and reproduction are the same. So it's flat. Oops, let's see. Oh, come on. It's totally flat. So stationary is a plateau. So death and reproduction are the same. So they're starting to die at the same rate that they're being reproduced. Okay, and then part D is actually, this is called decline. And in decline, death exceeds reproduction. So, so what's happening is you're running out of food. You might be running out of space, so they're crowded, but they're dying. Okay, and eventually they're just flatten out and they'll be all gone. So we have lag, is they're getting used to the environment that they're in. Log, they're growing like crazy, so they call it logarithmically they're growing, or exponentially, so every one is giving rise to two more, so it's growing like crazy. Okay, and then stationary phase is a plateau. Okay, so death is equal to reproduction, so you're starting to run out of food or run out of space. And then decline is death. Okay, so death exceeds reproduction, so they're running out of food, they're running out of nutrients, obviously, or they're running out of space. Okay. All right, and I have the same, whoops, wait, now I gotta get rid of that. I don't know what I did, but anyhow, here I just wrote it out here. It's the same thing that I just labeled on the graph. Okay, and that's that.